Okay, I'm actually covering also for Reza Zomorodi, who cannot turn up. So I'm incorporating his findings in a clinical study for meditators. So you'll find it somewhere there. So I've taken advantage of this and speak more, of the substantive stuff to teach you too. All right.
So you'll be speaking for about 40 minutes.
Yeah. Of like 35 and 5 for Q&A, but who's counting when you're not quite sure? Right. So I'm going to be speaking from the perspective of empirical evidence. Now you've heard a lot about the theory and coming up from, say molecular level or from quantum physics perspective. But I wasn't very sure about. I was kind of standing on a fence until this evidence came up. So I was trying to figure out, and you see my my little journey here. Now, photobiomodulation is really about delivering photons into your body and does something, modulating the biological system. But we focus on the brain, so we talk about the brain. And here is an example of a device that does it through the LEDs. Now, this is a field that is not quite well known, but people in our field is growing. They are biohackers, they are serious researchers, full of nerds, and I think the evidence is bearing up very strongly. Particularly, we get involved in other forms of brain stimulation too. So this has a very good foundation, and it is building. So I'm not going to go into the basic, very foundational mechanisms like what happens to the mitochondria, but you just have to bear in mind that foundationally it works on the mitochondria. If you're an expert in mitochondria, you know how that works out. But I want to go straight into what we're trying to do to see what it does to the brain. So we're doing this study. It's actually still going on. We finished recruiting. We're collecting data and analyzing data right now. But this is going to be on 43 or 45 subjects. Now we picked two irradiation spots with optic fiber delivering laser, one on the forehead, one to the nose. We have to use it this way with laser and optic fiber because they're compatible with the MRI scanner. And what do we see? Now, these are a lot of images. I just want to give you a feel that we used two different wavelengths, 808 nanometers, 1064 nanometers. They're both near-infrared light. And one is through the transcranial, one is intranasal, and this is what we see. The peak, and this is this is a composite of all the 43 subjects. The peak for what you see here to transcranial, the head, is 150 milliwatts per square centimeter, and intranasal. I just want you to note the difference because this is kind of important to us that it's only 9 milliwatts, about roughly 15, 20 times less power than putting one on the forehead. And generally, through fMRI imaging, you see more response from the brain in terms of blood oxygen level dependency. After 4 minutes, you can see the whole brain lighting up, and it's global, just 1 point, and the whole brain lights up. So this is just the takeaway from this one. Then we also did the EEG reading, real time again, with 256 electrodes. Now we can do this because, you have a lot of electrodes. You can put one on the forehead and one inside the nose, so it's not interfering with the LED electrodes. And this is what we see is all over the map, right? This is 808, 1064, two different wavelengths, but the difference is actually very significant. 808, this is should be intranasal. Should be transcranial. This is intranasal. No, this 10 hertz is 40 hertz, sorry. So we pulse at 10 hertz, we pulse at 40 hertz, alpha or gamma. This is only transcranial. We have intranasal, but we haven't fully analyzed the data yet. But again, look at the difference between two different wavelengths and two different pulse frequencies. If you're feeling like really overactive, you may want to go here, sorry, overactive, okay, you want to increase the delta slow wave, slow you down, or you feeling like you need to sleep, you're increasing delta and decreasing gamma, example. If you are, say, inattentive, 40 hertz, you want to maybe decrease the delta, the slow wave, and so on. So there are advantages in having all these many different characteristics, and we will find a way to combine them and deliver to the brain. And just to recap, the brain responds significantly to photobiomodulation. It is global. The wavelength matters, as I've showed you. Power density matters. The intranasal is most efficient. Why is that? Think about it. There's nothing between you and the brain, right? It's just nasal cavity. The olfactory bulb just sitting above it is part of the brain. And frequency really matters. We are using this as a basis to see what happens further. Now, you may have attended Earl Miller's talk, and I actually followed what he's been doing. Out of all this, he did a really good presentation, talking about the faster rhythm gammas as actually associated with bottom-up processing with alpha and beta, electrical field influence, and so on. What we want to move on from here is brain rhythms are not just byproducts of neural activity. They are critical in organizing it. So for a long time in our field, I've always been wondering, is this EEG that we see, is it just a byproduct of your physiological action? Actually, it's more. He's saying here that if you can influence the brainwave patterns, you can influence the deeper physiological actions of the neurons. So we did a study. I will say that Sanjay follows me later. He is doing a parallel study at Tucson with University of Arizona. We were doing this in Toronto, and we wanted to see, now that we know that photobiomodulation can influence the brain, say by frequency, what happens? This was a double-blind study, 20 subjects. It actually is not published yet. we are just discussing about publishing it. Very briefly, is 5 minutes rest. This is all taking the EEG readings, and 20 minutes of EEG and photobiomodulation. So meditation plus photobiomodulation, resting. So we collected the data, and we pulse here: 4 hertz, 10 hertz, 40 hertz, 120 hertz. And the sham, of course, PBM off, sham, and then there's control, just meditation. Later, Sanjay will present data on faster frequency, and that's important because we kind of stopped at 120 and saw this significance at 120 hertz. We saw more significance at a higher frequency than the slow ones. And we had EEG data, because 120 hertz with meditation and photobiomodulation is the significant combination. I just wanted to show the EEG. Actually, the EEG is very hard to interpret, to be honest. But what was significant is the primary endpoint that we use, which is the mystical questionnaire. So it actually, a mystical experience questionnaire, it measures mystical positive mood transcendence of time and space and ineffability. So that is primary point, and actually significant. That gave us very significant outcome for meditators when you measure it that way. So this is just a summary to say high frequency, showed higher impact, and it shows suppression of the low frequency power, like, this is delta. Now, let's look at how this is happening. And this is where I guess this conference comes in, and we will resonate with it. But what we also did was we did cell culture study on turbulence, and looking at optical density, we found that it controlled when we pulse with this wavelength at 10 hertz. What happens is the optical density decreased. So we see a depolymerization here shown in this chart. We actually published this already, and we were in collaboration with Jack Dudinsky in doing this at University of Alberta. Now, this is not published yet. Nobody's got a time to write this. But when we do different frequencies, the crossover point seems to be at 40 hertz, like gamma, So gamma means something to people, but when we do change this frequency to higher frequencies, the crossover point seems to be at 40 hertz, and as we increase the frequency, 120, as we go to 1,000 hertz, we see it goes the other way. There's repolymerization of the turbulence. Now this gets more interesting. So we wanted to see what's happening to the protein structures. This was published with, we did use the Raman spectroscopy. And I actually showed this to Anirban. Anirban said, oh, you didn't do it quite correctly, but you published it. And I'll use data what we have. He said it shouldn't look like spikes, but this was how we collected it, and we hope to do further studies, maybe with Anirban with another set of Raman spectroscopy. But the point is there is a shift from alpha helices to beta sheets. And the thing about it, why it might be important, actually, when you talk about the dipoles, and you talk about the aromatic rings and all, and the structure of the turbulence, they're actually made of helices, the helical. Beta sheets sometimes seen as an issue. Then it looks like you're transitioning towards creating these structures associated with deposit and plaques and beta amyloid and maybe neurofibrillary tangles. So if we can do this shift, I think it's good. The alpha helices is actually more robust than the betas. The beta strands lead to the beta sheet. So you actually want to see that for a more robust structure in say the microtubules. So that leads to other things, right? You're talking about microtubules being the foundation of a lot of other things you see that's related to quantum resonances and all that. Now just to summarize, so 10 hertz leads to transitioning. In the microtubule protein structures, 40 hertz is consolidating, polymerizing. As it goes faster, it seems to shift more and more towards that.
Can you explain the significance of polymerizing or depolymerizing tubules?
Okay, polymerizing is like aggregating. So the tubulins are like getting together. I think that's a simple way to put it, aggregating into a stronger structure that became microtubule. More questions we'll leave to Q&A. But if you have something that needs clarification, just let me know. Okay, seems to be a key to further understanding, to consciousness. And I was wondering, okay, why is this happening? So through deduction, I felt that there should be resonance somewhere, and there's a lot of discussion already in this conference about resonance. And so this is Stuart Hameroff's. I thought it's a really brilliant way of schematic presentation of what's the, as you go deeper and deeper, the structure of, structures that are related to neurons and microtubules and tubulins. So when I'm not sure I did a calculation. Okay, anyway, at 810 nanometer, you do this wave frequency, use the wave frequency equation, the frequency is 3.7 times 10 to the 14. So you go somewhere here, is not quite, so this 10 to 14, when we talk about the tryptophan absorption, that's 10 to the 15, that's pentahertz. So it's somewhere here. And, but we also have the other frequencies. Remember, with the meditators, it's at these frequencies, which is somewhere here, right? And so we're wondering why these meditators actually had this shift. I showed you 120 hertz. Actually, we did a lot more outside of this study, and we just put meditators through the different frequencies. They go through a jhana state. Some of you may have already experienced with our devices, or an altered state, a bliss state at a certain frequency, and it's usually above 40, but many are like when we found 120. I'll leave to Sanjay to talk more about that in his presentation later. So there seems to be a coupling, and I thought, okay, do we multiply this into that and give you a really high frequency? No, actually, these fast frequencies are nested into the slower one, and that is coupling. So with that specific frequency, there's like a consciousness leap, and Stuart Hameroff would say, the bing, and okay, it suggests there is, there's the OR in play, which earlier I said, okay, I'm not sure, but it seems to me as I'm shifting towards, using that as an explanation. Then deeper down is quantum coherence. So, now this is interesting. I prepared this first, and I sat through Earl Miller's talk yesterday, and he said, and there was a discussion, and he said the brain is like an analog computer. What we have here, we're talking about digital here, quantum. So I listed this hierarchy actually based on the amount of evidence that's out there. And there is far more evidence in the literature that supports that kind of work on the higher level, neural synchronization and neuroplasticity. And here we have a lot of discussion on microtubular resonance, electromagnetic field, quantum coherence. So that is more like this lower area. These are psychological evidence. There's some coupling, and biofields is not a lot supporting it. So we can break up into basically following his use of word analog and quantum here, or you might say digital. So let's cross that out, and you can actually connect them. So the question today was, how do we link these two, analog and digital? I think we've taken a step towards that, and photobiomodulation, which we used, seems to be drawing that out and connecting the two levels. That leads to this consciousness leap that we saw in the expert meditators. Now, so this is really nice, and always wanted to translate what we see in this, and I'm doing all this, basic research is trying to get a better set of parameters to go and translate into everyday use and help conditions like Alzheimer's disease. We are doing quite a lot. We are planning to do a Alzheimer's disease trial. We've talked to the FDA. We've talked to FDA for traumatic brain injury and a bunch of other things. I'm writing a paper now on brain fog and long COVID, which we have good data. But let's just go here for the time being. We have a very general, because of what we see in alpha and gamma effects based on our work and a combination of work, we are combining alpha and gamma in future clinical studies to try and treat Alzheimer's. So, but we want to try to personalize it because all brains are different. For one thing we know, everybody's brain is different, although you generally get good outcomes with a large percentage of people. And EEG is going to be the basis, and we are looking into incorporating artificial intelligence to tease out. I can talk a lot more outside of this meeting, but we see some really interesting things that can help us to do that. Now, this is under peer review, a pilot study done at St. Michael's Hospital in Toronto. They are part of the University of Toronto. There's a lot here, a lot of data, but we have looked into the effects of this device delivering gamma to 20 subjects. Is a double-blind sham-controlled study. And we looked at cognitive outcomes measured through the really significant one was through MMSE here, which is quite commonly used. Then we looked at serum mitochondrial metabolites and looked at the mitochondrial metabolism. Some of them are quite significant. I think the ratio between lactate and pyruvate is one that is pretty, I think, credible in looking at the effects of mitochondria on Alzheimer's. When I say Alzheimer's, I meant this is mild Alzheimer's, mild cognitive impairment. Then we looked at plasma inflammatory factors. We looked at the connectivity through fMRI, and we looked at even the volumetric change. So one thing the photobiomodulation does, also from our other work, is it seems to help the regrowth of brain cells, particularly in the hippocampal area. So this other study also did that. The main investigator actually used the MRI quite a bit. So these are quite significant numbers. It's still a pilot, and we are taking this to the next level, talking to the FDA and say, okay, the data seems to pan out. We have other earlier data, so we want to do a big clinical trial. Other further evidence of brain response, we got a lot more, but we want more answers, and we might be collaborating with some people here too. If you feel like you see something here, let's go find out more. I'll be happy to talk to you. We fund most of our basic research. Nobody does it. We are working with University of Calgary, looking at actually super radiance covered by Dr. Korean here too. Looking at the Jack Dynski group at Turin and Alberta on cultured neuronal signaling. We want to see the structural changes in microtubules with different interventions. So we have quite a lot more that we want to do. Now, we are also doing this, remember, we put people, we want to see what happens to the brain in real time in an MRI scanner. We are looking at collaborating with University of Toronto and Harvard in putting high-level meditators into the MRI scanner, real time, do the different frequencies and see what's happening to their brain. We don't know, it's never been done in real time. There is a big study done at Harvard by Matthew Satcher's group. using very high resolution T7 scanner and saw the changes in the brainstem, which look different from some of the earlier studies that have come up looking at the networks. So we will discover new things, and maybe we will one day, Sanjay and I were kind of working on, okay, let's do a shortcut and get people, people like me to be a good meditator and beyond, you know.
Yes, Lou, you're just doing it for yourself, basically.
Oh yeah, for myself, okay. For selfish reasons here, maybe we can help other people. So we have a summit coming up in Toronto. We are planning this, so you can register. I think we have physical and online presentations too. So go there, because we are interested to hear some of, it's not all about our technology. It's really, we find that as we do this, we are teasing out more understanding of what's happening to the brain and what's happening to the expert meditator's brain. So, and hopefully if this is good, we will continue every year. And we have a flyer somewhere, and you know, some of the speakers are actually very good scientists in that area. Now this is part of, we do a lot of collaboration. I'm putting up to thank a lot of people. What I presented to you is part of, we do in neurology, what we do in basic science, cellular structures and all that, and meditation. But we got some crazy, this method of brain stimulation is giving some pretty crazy outcomes in sports. For example, you know, American football college football players. Watch out, that paper by University of Utah has submitted for review now, showing also, you know, diffusion tensor imaging of the brain. I think that is objective measure. It's not just like I'm reporting, I'm getting better, and I'm scoring better in school and stuff like that. So we have a lot going on. And I'm writing paper now on brain fog in long COVID, which we had pretty significant outcomes. So these are a lot of people that we collaborate with, and of course our team at Vielight. Thank you very much.
We have 10 minutes for questions, so go ahead.
Okay. You
So one question.
Thank you for a fascinating talk. I have one question regarding the focus on microtubules. Within a neuron, other elements like the sodium-potassium pump, synaptic vesicles, kinesin, receptors, neurotransmitters, COM2, MAO, all have very critical functions in neuronal function. So assuming that the light, these light frequencies do affect neuronal function, have you looked at these other aspects of neuronal function, or are you just convinced it's just all microtubules and that's the end of the story?
Okay, we actually plan to look at the effect on neurons. We are planning to get neurons, stem cell-derived neurons, and see what happens to it. While we're doing this, you know, I always have this thing at the back of my mind. We're doing in vitro studies. It's not like the real thing inside your brain, right? So, so it's nice to see see what it does to the microtubules. Again, what we see is in like a petri dish. So to translate that, we really have to see what is happening to human brain, but it really helps us to do that. I can't answer that deep biochemistry. I'm not a biochemist on the different things. But the other thing we do, as part of this study, I didn't show it here, is we do studies on living cells. We had a discussion with some people. And the effects are actually very, very interesting. We can see changes in electrical properties on the living cells. They respond differently to different frequencies. And we found something really interesting. It's just like accidental, you know. If you're familiar with the properties of cancer cells, the activity is mainly glycolysis, burning of sugars, as opposed to oxidative phosphorylation, you know, usage of oxygen in the process. But we saw the shift. We actually, I think we submitted for publication or something. We saw the ratio shifting with this. It is quite a big thing. I'm not a cancer guy. I try to avoid going to cancer because it's so political, you know. I've written papers, people keep asking me to do it. But we are the only viable living cells we can get to do experiments on a petri dish are actually cancer cells. We are finding a lot of interesting things. We find, you know, we can suppress the activity. Your question there? This guy here.
Hello, thank you so much. I'm just wondering about the long COVID study and the brain fog. It seems to be your focus, and if you could potentially talk more about that, like what the particular features you're looking at, the interventions and the outcomes ultimately. Yeah, thank you. Okay, brain fog. We did a 12-week study, and we eventually recruited, we recruited a lot of people. We ended up analyzing 200 people. And we used, so there's different measures of cognition, you know. There is the NIH toolkit. We use the CRAYOS, which is a combination of different cognitive assessments. And basically, there is statistically significant in the improvement in cognition and memory. We also did a secondary. We did looked at the fatigue, which is the two major, the two major symptoms of long COVID is first it actually is fatigue, chronic fatigue, then brain fog, and then you have post-exertional malaise. You do something, you get too tired. So we are going to do another study because we manage brain fog, but we plan to look at the effect on chronic fatigue with vagus nerve stimulation. There is some evidence there. We'll see what happens. So that is in the being planned right now. I think there are a lot of people suffering from brain fog. I don't know whether by the time we've done the other one, everybody's recovered. But just do it. Like we actually did. We were successful in a acute COVID study with another device. It's approved by Health Canada, but acute COVID is not a thing, right? So that's over. Everything was successful, but nobody is interested in acute COVID now. But actually, it helps with the immune system. We got approval for upper respiratory tract infection by Health Canada because of how it's related to acute COVID. So this is my big chart of ecosystem. There's a lot going on. Yep.
Also, just in a population with mild cognitive impairment is the postmenopausal population, and has there any work in that? And
yes,
the second,
yes.
second question would also be, do you see any deleterious impacts, unintended consequences in these people?
Okay, I will start with a simple question, not much, negative side effect. In fact, we hardly anybody, nobody reports anything, nothing fatal. It's just, the main thing is it works on the mitochondria. It tries to reestablish homeostasis systemically. Now, perimenopause though is a big thing in our company right now, and we are actually literally planning it right now. We've got quite a prominent professor who is going to be part of the investigation. And it will be a combination of the brain stimulation and vagus nerve stimulation. The primary endpoint, what was the primary endpoint is mood disorder. And why do we not say brain fog? According to the expert, they say brain fog in perimenopause is kind of temporary until you hit menopause, post-menopause. That's his opinions. But the other secondary
endpoints are going to be sleep, which is a big factor, and hot flashes. Hot flashes is very interesting. It's hormonal, of course. We see contradicting outcomes in the literature, but let's do it anyway. And but the primary outcome is going to be mood disorder. Yep.
I have a question. Because of this brain-gut axis, what do you think about like stimulating gut as well?
Okay, gut axis has been the brain. Gut axis has been related to Parkinson's disease quite a lot. It seems many people believe that's where the origin is. There is a study being done by a group in Australia using photobiomodulation. They're doing this, but I think the direct gut action seems to be very unclear because of the amount of fat, between the skin and the gut. So they're looking more into the stimulation of the vagus nerve to reach the gut. And we might do that for Parkinson's. I think the gentleman was trying to ask questions. Anyone else? Do you want to ask? Don't know time? Okay.
Actually, I have a couple of questions for you, Lou.
Okay.
So you mentioned the vagus nerve stimulation. So do you have a vagus nerve stimulator?
Yeah, we do. Our vagus nerve stimulation device is nothing like what's out there, because out there, all of them use electrical stimulation. Either originally it was implant on the left side, and then they found that transcutaneous or non-invasive, just putting outside with electrical help. They're doing it in the ear. No one's ever done it with photobiomodulation because it's kind of non-intuitive, but it is kind of, for me, it does modulation of the body, so it can work. We pulse it at 100 hertz as opposed to the usual one. It does work. So we have a study that's coming up. In fact, they just signing a contract with Harvard, Massachusetts General Hospital to do that. They found in the preliminary testing, everybody does preliminary testing before they do a proper pilot study, and they did find a change in vagal tone. They put through induced stress, it helps. So we'll see. Actually, people who use this, they all say it works. And the thing is, you don't feel the tingling, and you have to use gel because you're not using electricity. Okay.
All right,
yeah.
last question. Have you used it? Do you use photobiomodulation yourself, and has your brain improved by using it?
That's my secret. I use it daily. Some form, we have a number of devices. And I would say that, my brain is sharper now than when I was, I was not a poor student. I was pretty good when I was in high school and college and all that. I pick things up a lot quicker now, actually. People know me, remember everything. I'm not a trained biochemist. I'm not a physicist. I pick this up.
All right, thank you.
Yeah, okay, thank you.
All right, now we have Antonia the Francesco with photobiomodulation and consciousness.
Thank you. Thank you. I tell a little secret for all you. My English is really terrible. So please open your heart and your mind. But I write. Okay. Thank you. Okay. I'm a Cam. It's not necessary. Thank you. I'm a chemist, a CEO and scientific director at IOMED. That is a center for integrative medicine where we use all the technologies that permit all of us to have a contact with the subquantic field or consciousness path. I teach in Italian vibrational medicine, color puncture, and chromotherapy. That is the old terms for photobiomodulation in the University of Rome, Tor Vergata. How? Interact with an holographic vision of universe in a medical center. How is possible this? I write everything so you may read. In an holographic vision of the universe and the human system, every single part has information about the whole. So we try to organize our work and our research in this vision. So theoretically, it's possible to remulate the information of present or karmic or genealogical memories. If that prevents the expression of a full life. The present moment contains all the time, and our perception of time is linked to the experience in living matter. And the experience is in this our living experience, but we have memories of experience in other windows, temporal windows. Some researcher worked on the family memories and how to regulate them in this experience. So, what we think in our center that the best human bioresonance frequency are sure our brain waves, what we produce when we think in our modus vivendi, in our life. But sometimes it's necessary use other bioresonance frequencies to hack limiting patterns of mind created by our mind, the way in which we think and live the experience. So, when this is speaking the language of cells, performing intelligence therapy, was said by Peter Mandel, that is the father of color puncture, is a mystic more than a scientist that worked on with the light frequency. And when Fritz Albert Popp demonstrates the existence of biophotons, so the light emitted by ourselves, what mysticians says of a lot of time, but just in the last years, this has been demonstrated by Fritz Albert Popp. So we use the human emission to make diagnosis and use light emitted from devices to give order in the system. The idea is that the chemical code follows an informational code. So we prefer work with the information directly. Information carried by specific frequencies of light activate specific biochemical process, like we have listened in most of the conference during this event. Understanding this mechanism opens new horizon in the health and welcome sector. Okay, what is biophotomodulation? I think that is already known and all of us. So I go. Okay. We in our center use a device that emits in the near infrared. In the beginning, when people arrive in our center, we make a measure of the energetic emission, and after use a device to optimize the performance, the mind performance, enhance the ability of creation, and biohacker the mammalian emotions, where mammalian emotions are not the right emotion for the human that want to create a new reality. The idea is that there is a place in our mind that some researchers have, when they isolate the people as not more fire and all the limited emotion, and maybe more free to create reality. After, we use color puncture, that is the now the best name will be photopuncture, but Peter Mandel, 40 years old, when worked on these systems, called chromopuncture. From photopuncture of Peter Mandel, we work on meridians and parallels of the traditional Chinese medicine on or in reflex zone. In this way, we work to bring order in the system through the light, and this has light lateral effect, the fact that people stay in health, stay better, some pathologies are reduced or there are no more. Each light frequency activates specific windows. For example, according to Peter Mandel's chromopuncture, orange frequency regulates the endocrine axis. Yellow affects the liver, gallbladder functional circuit. Red-orange works on kidney bladder circuit. Blue and violet on spleen, pancreas, stomach. Green on the intestine circuit. Violet on the heart. So every meridians of traditional medicine has the windows where we may interact. Peter Mandel that I say is a mystic rather than a scientist, has identified points on the body, like the chip on the body, that interact with amygdala and allow the integration and harmonization of non-coherent frequencies coming from experience of the present karmic or genealogical memories. So we are able to put order in a system where emotion don't permit to live the optimal situation, physical, emotional, and informational. In our center, we use the intuition to bring health and wellness in our patients. And we use to monitoring what happens, we use an instrument that is the energetic emission analysis. Okay, this is the old instrument made by Peter Mandel. That is this, the person who organize all this system. And this is the image, is a Kirlian picture of the finger. What I use in my medical center and used here some day ago is the BioWell GDV camera. That is a project of the Professor Konstantin Korokov of the University of St. Petersburg. We work with him in this field. So what we have, this is a picture of some years ago. What we have all the emission from which we have information about the system. So all organs, tissue, meridians of acupuncture. Have information about chakra. So we have a connection with Ayurvedic medicine, traditional Chinese medicine, and information about the states of energy, emotive pressure, balancement of the organ, with a lot of information to read and to which give an interpretation. We take only the finger of the hand, and the image that we obtain is like this that you see. After all the systems read and transform the irradiance. Okay, so we use this system before and after a treatment to know exactly what's happened in the system. Okay, I know that my time is finished, so I give thank you to you and okay.
We have time for some questions. Who has a question?
Thank you. You talked about how different colors of light have traditionally been associated with different organ systems.
Yes.
But then how did you decide on 810 nanometers for your stimulations?
Just the second part of your answer is not clear for me. Sorry.
You said you stimulated with
Yes.
810 nanometer light?
Ah, okay.
Why?
I simulate with the first device just 10 minutes because I use him is when begin the treatment to put the systems in the best condition to receive the color puncture treatment. The color puncture treatment is too fast because is enough just few seconds to have an answer. Immediately the system give an answer.
Okay. You reported that you measure pre and post treatment. Do you measure any patient-reported outcome measures of how the patient themselves report the effect?
Yes, this is the information for me, but I have the background of patient, and patients ask analysis and a lot of information, not only energetic. So we monitor them in this way because this is the way that I love. But there are all the analysis that confirm what happens. Thank you.
Okay, I want to try to help to answer the 810 nanometers. That device was developed by Paolo Cassano of Harvard. He's a friend of mine. Now, why do we use? I mentioned a lot about 810 nanometers or 1064. It's just historically it goes back to the days of the lasers, before LEDs came out. So a lot of work was done around 808 nanometer laser and 1064. There were computer modeling done. There's several computer modeling done to see what wavelength draws off the best effect from the brain. On balance, I'll say 810. We show that on fMRI generally, but I'm seeing different things now. 1064 seems okay. 810 is also in the action spectrum to activate the mitochondria through cytochrome C oxidase in the electron transport chain. The others, like 1064, does not. Actually, in that, it does another, another set of mechanisms. But generally, for the brain, it's very popular. Either it's 810 or when they use LED, it's 1070, which is close to 1064. Does that answer the question? Yeah.
Thank you.
Thank you, Antonia.
Thank you.
Thank you very much. All right. So next we have, that's me, Sanjay Manchanda, talking about supporting meditation with photobiomodulation. And this work was done in collaboration with Brian Lord, Erica Cook, and Jay Sanguinetti of the University of Arizona. And some disclosures. I'm an advisor in equity to a photobiomodulation and neurofeedback company, SenseAI. I'm a consultant with Vielight. Vielight funded this meditation study that I'm going to talk about. And I also have a couple of websites in the making, a Meditate with Light website and a Neuromatrix website. So I'm selling some photobiomodulation headsets for the purposes of meditation and health. I wanted to start by talking about just a couple of interesting mechanisms of infrared light. So one of the things that happens when infrared light strikes a cell membrane is that it actually affects the structured water at the membrane. So there's a thin layer, nanometers or micrometers layer of structured water at the membranes of cells, and the light affects it. It's already charged. The water is already tends to be charged and polarized, and the light increases the polarization. And it also heats up the water. And in this way, doing this, it actually activates heat and electrically activated and light-activated ion-gated channels. So that's one of the mechanisms by which infrared light works, along with the fact that it's absorbed by an enzyme. A therapeutic window is absorbed by cytochrome C oxidase, which is part of the respiration chain in the cell. So it's basically acts like food because it supplies electrons in the electron transport chain in the respiration cycle and creates ATP, the energy molecule, and reactive oxygen species in a small amount that beneficially signals the cell. So by supercharging mitochondria by these two mechanisms, it supports a healthy cell, and it actually also supports cell death of unhealthy cells. So just a couple of things about the structured water. When liquid water touches a strongly hydrophilic surface like proteins and cell membranes, the several hundred micrometers next to the surface reorders it into a quasi-crystalline form. And this water is very clear. It excludes salts from it. And it's like a microscopic bead. It carries a net negative charge, and it polarizes the cell. So this kind of structured water will absorb infrared light and near ultraviolet, and the absorbed energy can expand this exclusion zone further, and it creates a bioelectric effect from this absorption of light. So when you have infrared light beyond 950, it's the primary, there's some activity on cytochrome C oxidase, but as Lou was talking about, that when you go beyond 950, the major activation of infrared light is actually by the structured water. So this will expand the exclusion zone in the cell, create measurable millivolt potentials, and this electrical energy then becomes available to nearby proteins, and which activates ion-gated channels, and there's an influx of calcium and other beneficial minerals into the cell. So that's yet another way the light is beneficial to the cell function. And so there's ion channels that get activated by this, including heat. There's also some heat action here. So in terms of the activation of cytochrome C oxidase, typically what will happen is that part of the respiration cycle, we have nitric oxide that's caught up in the cytochrome C oxidase, and the light will release the nitric oxide from the cytochrome C oxidase where it's bound to copper and iron, and so that oxygen can come back in and the respiration is restored, and nitric oxide is released into the bloodstream, which further improves blood flow in that part of the body. So there's a number of benefits we talk about. Lou's been talking about it, so I'm not going to go into it. But all these benefits that these studies are showing in the various diseases and for brain function are because of all this beneficial signaling in the body, reduced inflammation, increased blood flow, increased antioxidants, anti-apoptosis, and reduced neurotoxicity and so on, increased angiogenesis. So our interest is more in state change and in pulsing the light. So what happens when you pulse the light? And we know that we have audio entrainment, we have visual entrainment that can happen. So what about transcranial infrared entrainment? Now Lou already showed some really great studies on pulsing a laser in the nose and on top of the forehead, and we saw all these changes in blood flow and how they varied depending on the wavelength of the light used, the infrared light used, and how much it was pulsed, whether it was 10 hertz or 40 hertz. And so there's these resonance effects that he was talking about, and there's also resonance effects not just at the microtubule level, but what I'm saying is they're also there at the ion channel level. And so each pulse briefly overpolarizes the mitochondrial membranes, increases ATP, and then when the pulse has the dark period, then there's a recovery. So for example, some studies show that 10 hertz or 100 hertz is better for neuroprotection in mice. This was done in Petri dishes. or even a 300 hertz is best for mitochondrial function in stem cells, which was done with human dental pulp stem cells in petri dishes, just playing around with the effects of frequency and light on cellular response. And we know that there's all these inner rhythms in the body. There's hemodynamic rhythms, there's cortical rhythms, there's ion channel rhythms. And when you have resonance with these rhythms, you will have a better effect maybe, or a different effect of the light than if you did not pulse it at all. And so this is another study by Vielight, originally done to show that pulsed biomodulation has a clear effect on the brain. So 20 minutes of 40 hertz pulses into the cranium using their Vielight neuro device showed an increase in the EEG of alpha, beta, and gamma. So we know that it's doing something. You know, this was a study he was talking about. I won't go into it. Pulsing a laser at 40 hertz, whether it's 808 nanometers or 1064 nanometers. And in this case, it was interesting that 1064 nanometers at 40 hertz produced the greatest and the most global increase in gamma power in the brain. This is another study done by MIT on the effects of 40 hertz. 40 hertz is a very interesting frequency for the body. So when in their research, when the light in the mice cages was pulsed 40 times a second, and these mice are bred to have Alzheimer's, then when they noticed that at that particular frequency, when the mice were sacrificed the next day, there was a decrease in the tau proteins and an activation of the microglia. So that 40 hertz itself, even visual and audio stimulation, what they discovered is generally beneficial for the brain and initiates garbage collection. So they have discovered in these, the researchers at MIT, they did a few studies on this, that increasing audio and visual 40 hertz power and synchrony in the brain improves cognition and memory, prevents neurodegeneration, and reduces tau and amyloid buildups. It stimulates the brain's glymphatic system. So this is independent of the positive effects of the light itself. The pulsing frequency itself is very beneficial. And so they have a genus project that's following up on this. What my interest was, this is the background, right? So what is my interest was, I'm a meditation teacher and a psychotherapist as well, and a biofeedback researcher. So I was using light to help people to have their brains recover. And then I thought, what about, stimulating the brain at a high frequency or various frequencies for meditation? I was already using brain stimulation technologies in magnetism as well as a current. But the light seemed particularly optimal for this purpose. So let's see what some of the thinking behind that would be. We already know that meditation states are somewhat related to EEG states in the brain. And we have been available lots of entrainment technologies that you can buy for a long time, decades, which do audio and visual stimulation, for example. And I'm also a biofeedback therapist, so where we're using biofeedback to change the EEG and correspondingly change the emotional mood as well as attention state of the client. Now, the question here was, instead of doing biofeedback or regular audiovisual entrainment, we are going to flash infrared light into the brain. And we want to affect the EEG and correspondingly help the person to change state. So the question is, what are optimal frequencies of stimulation to do that? How fast should we pulse, in what order, and what sequence? So I'm not going to go through the regular frequency band summary of EEG, but in general, I want to point out that when you're doing stimulation of the brain, whether it's current, magnetism, or whether you're even using neurofeedback, you will have an increase in arousal as you go higher. Oh, really? 5 minutes? Okay, I have a lot to talk about. Okay, so in general, as in a mindfulness state, there's a balance between low frequencies and high frequency states. You could say there's a balance between characteristics of meditation, like tranquility and calmness and energy. So one is stimulated by high frequencies, and the other is connected to low frequencies. I'm going to have to skip a lot. I was still going to talk a lot about the motivation of why I wanted to use gamma stimulation, but the basic idea is that can we stimulate gamma in the brain, and what is that going to do? And we know that gamma is implicated in experienced and long-term meditators. The long-term meditators have more gamma than beginning meditators, both at rest and in practice. When they practice, their gamma goes up. So being a long-term meditator, I wanted to see what I could do for myself. And what if I flash the light at high frequencies? And what we found was that it was surprisingly effective. So, and there's some evidence from using DMT and ayahuasca, which also reliably boost gamma frequencies. So what we found was that as we stimulated high frequencies, there's increased self-observation, equanimity, positive emotion, expansion, feelings of expansion, and openness. And there could be some overstimulation as well, some agitation and dissociation. And so we did a combination study with combining low frequency and high frequency stimulation. And I'm going to skip this one. So it was a single blind, just a pilot study with 20 meditators and using baseline active and sham stimulation, and pre and post EEG for each session with meditation measures using the Vielight NeuroPro device. And here's the meditation protocol we used, which is going from low frequencies to high frequencies, a total of 33 minutes, going from all the way from 1 hertz to 1,000 hertz. And using a 50% duty cycle and synchronizing it all over the brain. Another interesting thing is that when you synchronize stimulation all over the brain, then the brain can go into a simpler state. Normally, when you're in a thinking state, the brain is more desynchronized in various parts. But by maintaining synchrony through the brain, you actually make it harder to think. And people do report that. Oh, it's harder to think. It's both the high frequency stimulation as well as the rhythmic timing all over the brain. And so we had results on the MEQ scale, particularly in the transcendence aspect. So there's the baseline active in the middle and the sham condition. And the transcendence scale was more significant. And a fairly good effect size on the total score, the transcendence score, and the ineffability score. So there's something about higher frequencies which support transcendence, actually. The experience of transcendence and ineffability is what I also am finding subjectively. EEG analysis didn't show too many significant results, but there was an increase in the alpha, beta, and gamma frequencies. And there's something more we want to look at here, because one of the things I was talking, with someone about is that there is, we didn't have a specific meditation technique that we told people to do. They were focusing more on in general on the body, but not using a specific techniques will muddy up the EEG results considerably. Okay, so that's what I'm going to stop here. I'm going to take a couple of questions, or yes, go ahead.
One of the major problems with non-invasive neuromodulation is inter-individual variability.
Yeah.
Some people respond a lot, some people don't.
Yeah.
Do you have any approaches as to how you can personalize and maybe make it more effective for everyone, like with functional or structural connectivity data?
Yeah, exactly. I mean, there's another phase of the study we were planning to do. We're still thinking about that, which is that each meditator responds differently, and also there's an optimal frequency for each person that stabilizes them at for a certain time. So if you go over that, then there's discomfort in the body, and but if you go too low, they're not getting enough benefit. It's almost like the frequency indexes their level of openness in a certain way. So you want to find an optimal index for them. So for that, what is the EEG parameters we're going to look at? That's what we have to see. We have to do a better analysis of individual meditators. This was group analysis, but really we have to look at an individual's initial EEG, how they respond to stimulation, to then see, to actually, it's almost like you cannot do just an average analysis. And you have to do more individual analysis and get some patterns and their relationship to certain frequencies, which is what Reza is actually also trying to do. Yes.
So you said you helped people recover their brains. I'm wondering if you could share maybe the most impactful story you had with being someone who helps people.
Okay, this is not directly the well, there's two different stories. So one, if you're talking about cognitive performance, yeah, so, well, okay, we have so many stories. This is separate it's not so much the meditation study, but I can give you an example of how photobiomodulation can be very dramatic. A good friend of mine, his father is 95 years old, and he was losing his brain function, and he was saying that he's ready to go because his brain is very valuable to him. He has a great mind. And I said, why don't we try photobiomodulation? So we gave him a helmet that I sell and set him up, and we had him using it 2 hours a day. Now, normally we don't recommend using it 2 hours a day, but he was 95 and he's metabolically compromised. It would be too much for a younger person. And I set him up not with meditation frequencies, but with beta frequencies. So we programmed it for beta band frequencies. Beta band drops during meditation. But here's someone who's having a problem thinking, he's not interested in meditation. So we wanted to try and activate the beta band. And we ran the beta band frequencies with him for hours a day. And within 2 weeks, he was like, I feel so much better. I don't need to go anymore. So there's many stories like that. But another story in terms of the meditation would be people typically report more equanimity. So I have students who bought headsets from me, and we have a research group about 45 to 50 people. We share our results. Common reports are deeper equanimity in life, and also more emotions being freed up, and they come up to the surface, but there's more equanimity to handle them. Because this is not just a non-body-based technique. It actually opens up the whole body energy system. And so it's an embodied awakening kind of technology. So it will bring up old material to the surface, but it also helps you to deal with it. And it's all of this work with meditation devices is best done in the context of a system of understanding, a right view, and right other methods that are available that you can use to integrate whatever is happening with you. So it's not to be done in isolation. That's very important. Yeah. Final question.
I was just wondering, because one of the numbers you had in your special list was 320, and one of the numbers of the wavelength
Yeah.
you gave was at 850, or
Right.
which when I did a wavelength to frequency conversion on a pack and then brought it down,
Right.
it's 320. So I'm wondering if you have found extra efficacy when you get the wavelength to be an octave over the hertz.
Oh, that's interesting. I haven't thought about that, but thank you for that. I'll keep that in mind. Thank you all.
What is the cost of the price of the system?
So the cost is one headset for 3,000 and one for 5,000. Okay. So next we have. Okay, next we have Garret talking about PTSD and lucid dreaming.
All right, thank you. Hello, everyone. Thank you for being here. I'm excited to share with you the work we're doing at the Institute of Noetic Sciences. Hello? Pardon? I'm excited to share with you work we're doing at the Institute of Noetic Sciences in California, evaluating a workshop designed to help people with PTSD. So here's an outline of the talk. I will introduce the concept of REM therapy quickly. I'll talk about lucid dreams, some induction techniques that we could try here. Talk about healing lucid dreams, and I'll try to blaze through these 4 points so that we can focus on the workshop itself and the 2 studies that we've done. So first, the idea of REM therapy. REM therapy is something that can happen during REM sleep. You all know REM sleep is famous because of the rapid eye movements that occur during that time. This is where most of our narrative-based dreams occur. And as you know, the body is paralyzed during REM sleep because of the inhibition of the motor neurons. Also, there's an inhibition of neurotransmitters during REM sleep, so the fight or flight neurotransmitters are dampened. And a correlate of REM sleep is deactivation of the prefrontal cortex. So the prefrontal cortex is offline, basically the activity is down, and one of the functions of the prefrontal cortex is executive function, decision making, and also it's one of the areas of the brain where the sense of self is believed to reside. So this makes a little bit of sense if you think about when you're in a dream, your identity can change, you can turn into an animal or somebody else and you don't notice it much. So the theory of REM therapy is that because we have the neurotransmitters dampened, it allows the brain to decouple stressful emotions from the memories. So here's a cartoon describing that. So with multiple cycles of sleep, because we have the dampened fight or flight neurotransmitters down, you can have the memory and it becomes decoupled gradually from the physiological stress. Here's a paper if you want to look into that. So now what are lucid dreams? Lucid dreams are a dream when the dreamer wakes up, is aware that they are asleep in bed and they're having the dream. Sometimes can allow the dreamer to have influence the what's going on in the dream rather than just being carried along in the story. And about 1 in 5 people have these dreams just regularly, spontaneously. Could we have a show of hands of who has experienced a lucid dream? Wow. Okay, more than 1 in 5 in this audience. Not surprised. So, modern science started talking about this in 1913 when Frederick von Eden introduced the term. And in 1975, Keith Hearn was the first to demonstrate this in the laboratory. So he took advantage of the fact that the eyes are not included in this paralyzed state and made a deal with the dreamer that when you wake up in a dream, to send a signal, which was to move the eyes all the way to the left and all the way to the right and back and forth. And so this was a way to indicate to the researchers in the lab that they had woken up. Now this was part of his PhD thesis, but then in 2010, Stephen LaBerge at Stanford University did this and published it in a peer-reviewed journal and really unleashed all the modern research on lucid dreaming. Another landmark paper, about 100 years after von Eden introduced the term, was done an fMRI study that showed this prefrontal cortex lighting back up when lucidity was achieved. So remember, in REM sleep, the PFC is dampened, and then Stephen LaBerge had the people in the lab, when they became lucid, send the signal that they're awake, and this corresponded with the PFC lighting back up by fMRI. Okay, some induction techniques. So the 2 kind of categories. One are bedtime practices, like mental recitations during the hypnagogic state, or kind of hypnotizing yourself as you're falling to sleep. And one of them, for example, is and this is what I learned from the Carlos Castaneda books, and I was able to trigger my first lucid dream, is as you're going to sleep, you say to yourself, I will look at my hand in my dream tonight. I will look at my hand in my dream tonight. I will look at my hand in my dream tonight. And then, if you're lucky, you're in the dream, the story's going along, you happen to see your hand, and you remember, oh, I'm the one, I said I was going to look at my hand in my dream tonight. I'm the one that did that. I'm asleep, and that triggers the lucidity. Another type of practice are daytime practices, and my favorite one of those is reality testing. And we can do that now. Just try and move your finger through your palm. Now be careful if you've got long fingernails. Did anybody get it to go through? I'm still waiting for someone to just disappear when we do this. So I guess we're all here together, awake in this reality right now. That's just two examples. Now, a healing lucid dream. We talked about lucid dreaming and what is a healing lucid dream. This was the focus of the workshop. So healing lucid dreams basically is an intentional REM therapy. So remember that the PFT comes back online. So you've got your conductor, you can make these executive decisions while you have the dampened neurotransmitters, and this is the basis where you can basically influence the dreamscape to elicit the REM therapy.
Any chance decision can be made at that point when the PSC lights up?
Yes. Oh, well, I think that's it depends on your induction techniques, but it's hard to decide when that's going to happen.
Oh, I don't mean that. I mean, can the sleep dreamer make decision at that point?
Yes, when you gain lucidity, your decision-making processes as yourself are back online, yes.
And it will be observable, like it has consequences outside of the dream?
Outside of the dream? Yes, well, I'm not sure the question, but they're sending signals to the researchers in the lab with the eye signals. Yeah. Okay, so now the workshop that's designed to help these folks with experiencing PTSD. We work with the teacher, Charlie Morley, based out of London, who was trained in Tibetan practices for lucid dreaming. He's authorized to teach. And he had been doing these workshops designed specifically for veterans with PTSD. And he actually was awarded a Winston Churchill Fellowship for research because of this work. So we talked to Charlie and said, hey, there was all this anecdotal reports of these veterans experiencing great relief from their symptoms. And we just wanted to see if we could lightly put in some science to get some data to back up the anecdotes. The workshop is designed to be basically a 6-day intensive. It can be delivered in person, but we worked with him. It's being delivered via Zoom. It's altogether of the 6 days, it's about 22 hours of live instruction with Charlie. And here are some of the components of the curriculum. So firstly, he just talked about understanding sleep better, sleep hygiene, and the neurobiology of sleep, neurobiology of dreaming. Taught them deep relaxation processes, including yoga nidra. Techniques for dream recall. One of the first things he emphasized was that just remembering your dreams, recalling your dreams, is the first step to having more lucid dreams. Then, of course, techniques to induce lucid dreaming, the two that we just talked about, and many more. Dream planning sessions, so having intent to what they're going to do when they wake up in the dream, what are they going to do? And one of the aspects that was interesting to me is he said that people are more able to have lucid dreams when they have a real need, when there's something they want to achieve there, rather than just out of a curiosity. And in these cases, everybody really wanted to experience healing from PTSD, so they were very motivated. And then dream sharing. So every morning, there would be basically group discussion, people talking about their dreams, either lucid or non-lucid, and sharing that. There was also what's called Wake Back to Bed, which is having people set an alarm to wake up periodically through the night to perhaps help catch dreaming or write down the dreams they were having and then reinforce their induction techniques as they were going back to sleep again. So it increased the number of times that they had the chance to try to catch these dreams. There was homework between the sessions, so the live sessions, and then during the day, they had homework. So like the reality testing, more relaxation. And there was a psychotherapist present at all times throughout the workshops, both in the live sessions and also available in between sessions. And the goal, the overarching goal of this was to transform the trauma through the dream work. So as part of their dream plans was to transform their trauma. I just have a couple examples of what would be part of the dream plan. So for example, they could wake up in the dream and just call out to the dream, call out for healing. Some people would say they want to meet their inner child. And another example is just more general, is just asking, what can you teach me? Just being open to whatever the dream can teach them. So now that we first did a study, a pilot study, and this involved everybody with PTSD coming in, chronic PTSD, having been resistant to other therapies. In our study, there were veterans, but there were also non-veterans, just general other lifetime traumas. We had 49 participants. The primary outcomes were a standardized measure for PTSD symptoms, called the PCL-5, a nightmare experience scale, and positive and negative affect. We had others as well, but to make it brief, we'll just focus on these. So in the morning, there were questionnaires. Wake up, go online, answer the question, did you have a lucid dream or not? And then there was a series of questions. It's also a standardized lucidity scale that measured different levels of lucidity. Ask questions like, did you break any laws of physics? Did you remember your body was in bed? And then importantly for our study, we had a question, did you remember your dream plan? And this was our criterion to identify who had a healing lucid dream and who didn't. So for the pilot, we had 76% of the participants achieving lucidity within that 6 days, which was pretty remarkable. And the PTSD levels, these are the average of everybody coming in, was well above this threshold for PTSD, and immediately following, they were all below, and this was maintained at a month follow-up. I'm going to just quickly go. This was published in 2003, and it allowed us to get funding to do a larger study, a randomized control, so I'm going to skip to that now. This study, we basically doubled the number of participants, and after they were enrolled, they were randomized to either a waitlist control or the active workshop. And same workshop, same teachers, same measures. So just to be clear, in the waitlist control, they just had to wait. We collected all the measures from them during that time. Once we collected the final measures after that month follow-up, then they were offered the workshop so they could have their benefit. The results, in this case, we had 50% of the participants had at least one lucid dream during the workshop period in both groups. So this was a little bit of a surprise. It reveals a limitation of the study, is that when they recruited, everybody knows that this is a healing lucid dream study. They all want to have experience help and relief. And I think some of the people just couldn't wait. They just ended up on their own having lucid dreams. So that is indicates that what Charlie would say, that just even talking about lucid dreaming during your day can trigger these spontaneous events. So all of you might have one tonight just because of listening to this talk. In terms of healing lucid dreams, of the people that did get lucid, we did have a difference between, in the workshop, 63% of them remembered their healing plan, and in a control group, they just figured that themselves they had a healing experience, so they reported that as well. In terms of the outcomes, at baseline, there was no difference between our PTSD measure, which was great, but then there was a difference between the workshop and the control group significantly at both of the time points. This is what the data looks like, very much corroborating the pilot. Again, here's this threshold that all the workshop participants fell below. And this is the kind of effect that you would see with other approaches like CBT. Of course, this is just in 6 days, so it's faster. It's highly accessible, so only 8% were non-responsive as compared to about 50% with standard treatments. And the fact that it is remote is actually notable because for PTSD, there's some stigma, and it's helpful for some folks to be able to do this without going into a clinic. They can do it from their home. In terms of nightmare, there were no difference between the groups in the beginning. It did change at the follow-up. We didn't see a significant effect immediately after, but at the follow-up there was. Quickly, here's some notes on the statistics. The data were not normal, but our postdoc analysis did confirm our pre-registered test with improvements for both PTSD symptoms and nightmares. Our positive and negative affect, we didn't see any change in the positive, but we did in the negative at both the time points. And so just quickly, what are the mechanisms? Since the lucidity didn't, we had the measures of the depth of lucidity, it did not correspond with how much relief from symptoms. So the implication is it's not just the lucid dreaming, but more the process of engaging with this idea that you can heal trauma through dream work and pay attention to your dreams. So there were many workshop elements that overlap with other practices, for example, the group therapy. Another idea is that just learning about and actively attempting trauma transformation through dream work may restore a sense of agency that's lost in PTSD. And accessing the subconscious through remembering more dreams and dream sharing may reduce the disempowering impact of trauma. So I want to thank my team, Tadas Stumbries at the University of Vilnius in Lithuania, and the gang at the Institute of Noetic Sciences, Satara Tadeo, Cedric Kennard, Arno Delorme, Michael Kriesman, and Helene Wabe. And we thank our funder, the John Sperling Foundation, and all of you for your attention. Thank you. Any questions?
Hi, thank you very much. I'm concerned about one of the typical symptoms of strong PTSD, which is insomnia. And the very few occasions on which this person arrives and falls asleep by exhaustion or whatever, it's the night terrors, which can be repeatedly the same once and again. So how can you deal with people who are in this condition who can barely sleep and who have the only one recurrent dream whenever they have one?
Yeah, so two parts to that. One, just to be clear, for this intensive workshop, it was actually not helpful. It was a lot of sleep disruption for this, but this wouldn't be the way that would be applied during your average life. So the idea of night terrors, I guess the idea is hopefully with healing from this process, those problems would diminish. Particularly with one of the key teachings from Charlie Morley in this is that when you have a nightmare, this isn't your subconscious giving you an opportunity to integrate trauma. So if you become lucid in a nightmare, you want to try to hold on to it as long as you can, because if you wake up or you run away, you lose that chance to integrate that trauma. And that's supposedly one of the reasons why you don't have recurring happy dreams. You have the reason that you have recurring nightmares is because you have yet to resolve that trauma. So, but it's a very good point that insomnia is problematic. So, it just might not be, depending on the person, it might not be the best method if it is going to exacerbate the sleep problems. It's a good point. Thank you.
Okay, my question is, does being lucid affect rest or the natural course of sleep? I mean, due to the prefrontal cortex activation, does it affect to the natural course of sleeping, or maybe not?
Does lucid dreaming affect the natural course of sleeping?
Yes, because I think in REM sleep, the prefrontal cortex is not activated.
Right.
But during lucid dreaming, yes.
Right, so in REM sleep, normally the prefrontal cortex is offline, right? So just to be clear, when you wake up inside the lucid dream, you still stay in REM state. You're still asleep. You're just like totally out. This is the last phase of sleep. It's a unique state. It's been called a hybrid state, but you still do stay down in REM sleep until you wake.
You're resting in a normal. There's no difference in the rest, we can say. The quality is the same, or?
Yes. Well, overall sleep quality, the experience of it can be less rest. You know, it doesn't affect the deep sleep, but you know, sometimes it depends. And if you wake up out of it, you're so excited and you can't go back to sleep. So it can have an effect on overall rest, yes.
Okay, thank you.
Just curious, in the follow-up study, you said that you followed up the patients for a month after the stopping of treatment. I'm just curious, is this a cure, or does it something that has to be applied continually, the lucid dream and play
Yes.
with REM?
So the longest follow-up we had was 1 month follow-up. And so the measures were maintained at that point. Beyond 1 month, we don't have any data on that.
And is in PTSD therapies like CBT you mentioned, is this something that people continually have to participate in, or is it considered a cure? Is there a cure for PTSD, or is there just a,
I don't know that it's a cure. I would say that the people, just from listening to the reactions of all the folks and being in contact with them afterwards, it's something that they intend to continue to do throughout their lifetime.
And final question, and just your opinion, there's an emerging therapy of psychedelics for PTSD, and I'm curious what you think about that.
I think this is very similar to psychedelics in terms of the personal experience of it. It's hyper real. It's very much like a psychedelic trip. The one key difference is that this you can do in your own bed. You don't need to find a guru. You don't need to take some of the risks associated with it. Charlie talks about the subconscious. You're basically accessing your subconscious mind in this case, and you're doing it in this natural way. And the subconscious mind, its job is to protect you. This one kind of story about that's related. Some of the dream plans, an example of how the subconscious mind is protecting you. So there were cases where people would have a dream plan, like I want to meet, I want to, meet some aggressor from my past, or even meet my inner child. There are occasions where then a dream figure will come and say, you're not ready yet. So it's kind of a self-regulated therapy. So my personal opinion is it's a little bit safer than psychedelics, although it's kind of the same mechanism. Thank you.
Do you know EDMR? Eye
Yes.
movement?
Yes.
So how does it relate to that? I mean, EDMR was actually invented here in Barcelona.
Oh, yeah.
The doctors.
I think that one actual similarity is this decoupling of neural circuits.
Yeah,
so
exactly.
kind of the memories that are locked up there, this is an opportunity, like the REM therapy.
Yeah.
Whether it's lucid or not, I think just healing in dreams probably uses some of the same mechanisms.
But EDMR is probably easier, no?
It is easier, yes, absolutely, because
And it's very structured interviews and everything.
Yeah.
I don't know how much it is known. I know it because I have a friend who is a medical doctor who uses that.
Yeah, I think lucid dreaming, it is not easy to do sometimes. For some people, it's much easier, but that's why there's these induction techniques to practice.
Okay, thank you.
Thank you.
Thank you for a great talk. Oh, you have a question? Go ahead.
Very quick. No, you mentioned that in both groups, about 50% had at least one lucid dream, but then the effect of the non-control group went down below the threshold of PTSD measure. Like at some point you showed these two lines, the blue and the red. So it means that the lucid dreams of the control group were not as effective as the other. What's the difference between the?
So there were some folks in the control group that did have a lucid dream during the time, right? But there was a difference in the amounts. There was much less.
Okay.
Much less of them, yeah.
So
but it's just important that lucidity wasn't required to have this symptom relief. So just more than just a lucid dream. Thank you.
Next, we have Mihir Nath talking about heartbeat-evoked potentials as a neural marker of meditative depth.
I hope you're not falling asleep just yet after such a soothing presentation about dreams. So in this talk, we'll explore something at the intersection of mind, brain, and meditation. How the heartbeat, one of the fundamental rhythms of life, might actually serve as a neural compass for meditation. So our main goal with this project is to help any individual in achieving deep meditative states. This goal takes on a particular urgency when we consider the fact that for the people who would potentially benefit the most from a practice like meditation, for them it's usually the most difficult to start and maintain a practice. For people who are suffering from anxiety, depression, chronic pain, etc., it's very hard for them to actually follow the simple instruction of sit and observe your breath. So in this effort, the first sort of North Star for us was to create a real-time distance metric that can serve as sort of a qualia compass for neurotech-aided meditation. And as a step one, we decided to look at Vipassana practice and specifically look at the neurophysiological underpinnings of meditative depth. So here we're not talking about comparing mind wandering versus meditation, but looking at meditation itself within a session, trying to find more nuanced moment-to-moment shifts in meditation depths and how the different neurophysiological correlates might be able to track these nuanced changes. We also want our approaches to be personalized, to be able to serve as many people as we can, and we want pragmatic solutions that can be possibly scalable, easy to use. We want to move beyond 64-channel EEG caps to possibly consumer-grade devices. So this is sort of the experimental setup for this. You can see Nico, who's sitting here, meditating. So in this experiment, we had two main experimental blocks. The subjects we recruited were expert Vipassana practitioners with more than 10 years of practice. And they were essentially asked to meditate, do their normal practice, and report their depth of meditation with this clicker that you can see on the corner, which was on their hand. So we had two experimental blocks. In one of them, we used traditional sort of probing method where every 3 to 4 minutes they would be asked through an auditory probe, please rate the depth of your meditation on a scale of 1 to 5. And here, this 1 to 5 scale is a personal metric where 1 would be the most sort of initial beginning stage, shallower stage of meditation for them, and 5 would be their personal deepest state. Right. So in the other experimental block, we used a novel reporting method that we call spontaneous emergence, where the instruction that was given to the practitioner was to just, we asked them to observe whenever they would feel a sort of shift in their meditation depth that was significant, and then sort of voluntarily they would report the depth. And we found that even with that approach, we got as many reports as the other approach, and this is less intrusive. Right, so after the data collection, we had 42 expert Vipassana practitioners. The data included 64-channel EEG with different physiological markers, including ECG. After the data cleaning, pre-processing, we continued with 56 total sessions. Everyone had, one thing I forgot to mention, everyone had 2 sessions, 2 weeks apart. So yeah, we took 56 sessions. Right. So this was the first analysis effort that was done with this data in collaboration with Intheon. We tried different machine learning approaches to see if we can actually classify, predict depth of meditation just by looking at EEG. This is sort of one of our most most high performance results that we saw from the machine learning approaches. This is a whole list of different approaches we tried. And so what stood out to us was that a deep neural network based classification seems to perform the best. For a shallow versus deep classification where we sort of club depth 1 and 2 together and 4 and 5 together, we found about 81% accuracy in classification. It's pretty good, but not the best. Another thing was that the more classic standard EEG features like channel level activity, default mode network activity, spectral power did not really capture the sort of nuanced changes that would correspond to varying meditative depths. So that was interesting. And then sort of this got us thinking what other ways we can sort of parse this data, figure out other methods to differentiate between these sort of nuanced changes within meditation depth. And in that sort of effort, one thing that we considered was the role of interoception in meditation, meditative development. I really like this quote from this paper. It says, "The most intimate, unique, unchanging features of ourselves seem to be those which are quite literally closest to our heart." And this is a theme that is echoed across different cultures, different times. We sort of intuitively, when we think about ourselves, we point to our heart, and this sort of sense of the self residing in this sort of heart center is prevalent, this idea. Broadening across from the heart to broadly the body in general, we have the gut, we have the lungs, respiration. Many meditative practices involve deliberate attention to these bodily sensations, trying to refine our attention of these internal bodily sensation streams. Potentially, meditation sharpens our sensitivity and also our equanimity to these internal signals, and possibly this is one of the foundations of meditative development. Right, so this is a central key player in this study, Heartbeat-Evoked potentials. It's a real-time index of how strongly the brain is sort of tuned into the body. It's in the interoception literature, it's being put forth as an objective marker for interoception. How it's measured is that we look at the ECG signal. We can see there, it's sort of like the classic ECG signal, and then the most sharp peak there is called the R peak. It relates to the contractions of the heart. And what we do is that we run a traditional event-related potential analysis. If you are familiar with ERP analysis, how it works is that we have some sort of stimulus, usually external stimulus, and we use that as our 0-point onset, and then we look at the corresponding neural changes. But here, the sort of beauty of this approach is that we don't need an external stimulus. We have this sort of rhythm of life that gives us this stimulus that then we can look at the response in the brain. So how the analysis is done is that we create these small sort of segments of the data centered around the heartbeat. So we take about 200 milliseconds as a baseline, and then the 0 point is the heartbeat, and then 800 millisecond more. So we have these small data windows of 1 second, and then we average across thousands and thousands of individual epochs, we call them, sort of the segments of the data. And then we average across all the trials to get a smooth sort of response. Now, for our experiment, we did this for the different depths. So we had, these are the results, and here we crop this sort of part of the data. So this is where the depth report was done. This was the previous depth report, so we leave this 10-second margin after a depth report, and then we start sort of collecting the data. And then we also leave 15 seconds previous to the current report to make sure that we are not picking up on mind-wandering signals or maybe motor signals, because there would be a motor intention before pressing the clicker. So we wanted to make sure we ignore any confounding signals. Right. So these are the main results. We found that lots of channels show variations in the heartbeat-evoked neural response across meditation depth. The channel C3 stood out. It showed the strongest effects. And then if you zoom into this channel C3, here, so the 0 point is the heartbeat, and then we see before the heartbeat, it's flat. There is no, not much happening. After the heartbeat, about 150 milliseconds later, there seems to be a response to the heartbeat. And what we see is that the line in blue is, it relates to depth 1. It has the least response to the heartbeat. So the shallowest meditation state responds the least to the heart. Then we have yellow, which is depth 2. It's slightly stronger. Then green, 3, stronger. Red, stronger. And then at the end, we have depth 5. So it seems like and then in the other plot, we basically calculated the average EEG amplitude in this sort of time window of significant variation. And we see this beautiful stepwise increase across the different meditative depth. And it seems like as one goes more and more deep into their practice, their heart, their mind literally becomes more tuned to the heart and possibly all internal signals. So here we sort of combine reports 1 and 2 together, 4 and 5 together to sort of look at just shallow versus deep, low versus high comparison. And we wanted to somehow look at how this sort of physiological variation in heartbeat-evoked neural response might relate to some sort of subjective outcomes. So in the study, we also collected self-report questionnaire data that relate to mindfulness, mood outcomes, and other sort of measures of self-transcendence. And what we found was that we calculated this measure that we call HEP range. So it's essentially for each individual, how much range, how much variation do they show in their HEP response when they go from a shallow to a deeper state. So for some people, the range is higher, for some people, it's lower. And it seems like for the people who show a higher variation in the HEP range, they have less mood disturbance, which is a different way of saying they have better positive mood after the session. They show a lower sense of personal self, ego, and they also experience a higher decentering, higher sense of transpersonal self. So possibly more non-dual experiences also seem to track with this HEP variation. Now, to talk about the neuroanatomical significance of some of these findings, the channel C3 is above the left sensory motor cortex. The sensory motor cortex integrates signals from both bodily sensations as well as interoceptive signals. It receives interoceptive inputs via the thalamic relays and insular connections. The left lateralization is more puzzling. One possibility is that in the literature, it's been put forth that the parasympathetic control of the heart is predominantly lateralized to the left hemisphere, so it might be related to that. But another more parsimonious explanation could be some sort of interoception, motor integration, because C3 also overlays the part of the motor cortex that's responsible for controlling the right hand. and the participant mostly held the clicker in their right hand. So even though we made sure that we're not picking up on motor signals by excluding 15 seconds before the depth report, maybe this, because during the session there is a cyclic reporting via the right hand, there is some sort of leftover tracer print of the motor engagement and the sort of heart evoked response, and they somehow come together to give us this beautiful graph. So looking at it from a theoretical perspective, Ruben is sitting here. He was one of the sort of originators of this sort of interpretation. The theory of predictive processing says that the brain is constantly generating predictions about the sensory, different sensory signals that it's receiving, both internal and external. Deeper meditation potentially increases the precision weights or importance of interoceptive signals. So if that is the case, then during a higher interoceptive precision weighting, we would expect to see an amplified response of interoceptive signals, and heartbeat would also be one of those signals. And as one becomes more and more finely tuned to internal rhythms, there would be a down regulation of external distractions because you're putting more weight to internal signals, and this sort of also relates to some traditional ideas about sort of going back to the cave of the heart, etc. Our findings also echo the sentiments across spiritual traditions, especially in Buddhism, Hinduism, Taoism. In Sanskrit, we have words like the Hridaya Chakra, which is the heart center. It's considered the sort of bridge between lower and higher consciousness. Upanishads talk about the heart as the seat of the Atma, the true self. There's this quote, in the city of Brahman is a secret dwelling, the lotus of the heart. In Buddhism also you have similar ideas. Chitta is the word for heart-mind, the knowing quality of consciousness. In Taoism you have this character, Shin, I'm not sure if I'm pronouncing it correctly. It's a single character that means both the mind and the heart. So we have these ideas all over the world that talk about the heart-mind unity, especially in the context of contemplative practice, spiritual development, and our work potentially somehow relates to that. So we started this work because we wanted to create more accessible tools for making these peak meditative states more accessible to people. And neurofeedback is one of those approaches. We believe HEPs potentially can serve as a much more effective target for neurofeedback. Traditional meditation neurofeedback markers like frontal midline theta, alpha theta ratio, these target more indirect downstream correlates in our opinion. HEP potentially targets a core mechanism of meditation, which is interoception. There is another advantage, which is that every time your heart beats, you get a data point. So we have a very high temporal resolution, which potentially unlocks a much more nuanced, fine-grained moment-to-moment tracking of the depth. It also doesn't require any external stimulus or task, so it's much less intrusive, which is good. It's inherently personalized to the individual's heart-brain interaction. So another thing is it only requires a single channel EEG potentially coupled with ECG. It's potentially much more cost-effective and scalable. So this is a sneak peek into our neurofeedback design that we are working on. We are using audio accompaniment where there would be two different auditory tracks. One of them would be pleasant, high valence inducing, and the other one would be potentially more like not very pleasant to hear. And there would be a crossfade factor that sort of prioritizes one track over the other. And when the meditator would start, it would start from a neutral ground, and as they go into a more deeper interoceptive awareness, the crossfade would shift more towards the more pleasant music, auditory feedback, and we believe this could potentially create a positive reinforcement loop where the sort of hearing pleasant sound would first they would be able to identify the mental move that allowed them to access that pleasant sound, and then they would sort of keep repeating on that direction. We also want to use virtual reality, where we'll have a visual scape that would sort of adapt to their real-time AGP metrics, and it'll get clearer and clearer with some sort of visual feedback. All right. So this is the big team that made this work possible. Ruben is also part of this effort. He joined this paper recently, bringing in his theoretical expertise. And this work was funded by the Tiny Blue Dot Foundation. Thank you so much.
Hey, Mihir. Really, really cool work, you and team. I have 2-3 questions. The one that sticks out most is, a couple slides back, you have the 5 meditation conditions, and there's 5 bars of the heartbeat-evoked response. And to me, it seems like that in the, so meditation 1 is the least depth, and 5 is the strongest depth, right?
Yeah.
So what this shows to me is that in condition 1, you actually have the strongest response in the ERP. It's a negative response, but negative means actually the presence of response.
Yeah.
So I think the interesting thing here is that as depth decreases, you have a reduction of the response, and then you have a reversal of the response.
Yeah, some sort of criticality. Yeah.
And so I think there's something really interesting where, yeah, like this plot just does
right. That's
very different.
Yeah, that's a good way of looking at it. Any hypothesis about what might be happening.
And then the other question that I have for the neurofeedback, while evoked responses kind of have this really beautiful kind of time-resolved quality, you only get those shapes if you average over many samples.
Yeah.
And I'm curious if
it won't be as high sampling rate as each heartbeat. We'll probably have to average across like 10 or more to get like a more smooth response.
And what I'm curious about is, did you do any analysis to see how the accuracy of the classifier drops if you reduce the numbers? That would be something I would love to see in the next iteration of
So
this
we
work.
just got a grant for developing a real-time neurofeedback with this. And the first thing would be to actually implement the real-time HEP stuff, which is in the works. Yeah.
Love that.
Yep.
Thank you.
Thanks, Mihir. Great talk.
Thank you.
Could you remind me again what kind of Vipassana practitioners you had? Was it
Goenka.
So the meditation was also body scanning?
Yeah.
Okay, I'm curious, what do you think would happen if you would use different practitioners who maybe don't focus on the body? And derivative of that, for instance, when we think about the jhanas, and if you go into the formless states, what do you think would happen with that metric as a
right?
metric?
I think it can go either way, because even if you don't explicitly train bodily attention across different conditions, I think you become more embodied, like somehow, like maybe not first order, second order effects. So it could, we might see like a similar effect across different practices, but it also might not. So it's an empirical question, would be very interesting to explore.
Thank you. Yeah, it makes me think if the body drops off. If
You go whoosh, flat.
I think something that might explain the negative finding there could be intracranial pressure related to breathing. If you postulate that intra-abdominal pressure increases with diaphragmatic breathing, that increases as the meditation progresses, and that translates into increased intracranial pressure. And that will cause the heart evoked potential to be transmitted more strongly into the brain?
Interesting. I'll think about it.
I have a question. We don't typically have the meditator himself in the room with us. And I want to ask you, were you bothered or impacted at all with having to click the clicker? Yeah, you. How did? Thank you so much.
Thank you. About the clicking, I was wondering, isn't it more uncomfortable when you're in a deep state to click 5 times? Isn't it easier to click 5 times when you're in the surface state and more distractible? And then, you know, I'm in the fifth lower state, and you click 5 times. And when you're in a very deep state, you just click 1 because it's less uncomfortable. Could that make any sense methodologically?
I think Nico should answer this. He designed the study.
I may have missed it. I was just curious, in your analysis of the heartbeat.
Yep.
Did you use heart rate variability to examine if there was any relationship?
Not yet. It's on our to-do list.
Okay.
Thank you.